unfortunately become ill, but can provide her presentation anyway. And the talk title is about prophages. What are they doing in the school?
That's turn off the share screen.
I think it's on the YouTube link on here. We have a link to that.
Yeah, so you
find it, she'll send it to me.
Yeah. I think given that we are running a bit late, then I would say that we, I will encourage you to contact Ella with questions subsequently, just directly also because she has difficulty in answering questions directly. So we'll hopefully have her talk and then wait for the day.
Yeah. Is there something you're saying? I just said the name chat. Okay.
Any answer really?
Okay, that's great.
Quite slightly. Got my Google account. Need to sign this.
Okay.
What are they doing in that genome? So this is our sort of standard living, and this is a representation of the lysogenic pathway. And I'm particularly going to be talking about what these prophages can and do in this lysogenic cycle, because I think it's an area that is very understudied, and we are only just beginning to change our opinions on what happens in this place. I'd like to convince you that these prophages are very much involved in the biology of their host, is my belief. So bacteriophages, we all know, are viruses that infect their bacteria. They are fantastic because they're mediators of transduction, they're pathogens of bacteria, and they're drivers of evolution. We don't have to convince anyone in this room of that. But I'd like to remind you all that after genome sequencing, we have discovered that most bacterial pathogens carry integrated prophages. Some of these integrated prophages carry identifiable toxins, and that has been very interesting to learn that bacteriophage toxins can have a major impact on bacterial virulence. But still, most integrated prophages don't carry toxins. Yet we find that parts of the genomes of integrated prophages are conserved across strains, species, and beyond. And what we know from biology is that conservation normally implies important function. But my feeling is that for prophages, this conservation of function has been not given the attention that it needs, and that there is certainly much more to learn from prophages than we currently understand now. So I began work by studying Shigatoxin phages. This was an example of the first phage that I worked on that infected an E. coli K12 strain. And this little phage here is known as 24B. In 1982, the E. coli serotype was responsible for Shigatoxin-mediated disease was E. coli 0157H7. It didn't take many years for around 500 serotypes, though, to be associated with being capable of causing Shigatoxin-mediated disease. And it was clear that it was the movement of these small E. coli phages around E. coli and E. coli-related organisms that was responsible for the huge expansion of the host range of these phages. What became an interesting question to ask was, why? Why is this being disseminated so quickly? It was as if there was selective pressure driving the acquisition of these prophages, but there was no known benefit to carrying a prophage. And there was certainly no known benefit of carrying Shigatoxin. Shigatoxin causes disease in man. It's not responsible for disease in some of the ruminant hosts that are associated with carriage of Shigatoxin. So it was very hard to understand what that selective pressure could be. We moved on to using RNA-seq, where we could control the stability of the lysogen by the way that we grew them, and we could also force the lysogen and also the naive cell to undergo a stress response from induction, where we could look at all of the genes that are expressed in induction. So by doing RNA-seq, we looked at the expression of every gene across the chromosome under set growth conditions, and we could see the presence of the prophage within the genome of the E. coli cell. You can also see that there's a sort of distinct overall stress response going on. You can see that there is also a difference between the lysogen cell and the naive cell, and that these are the regions where the phage genes lie. We can also see significant differences in the lysogen gene expression that are quite different from the gene expression that we see from the reduced lysogen. So we had a way of reading changes in the host as a response of prophage carriage. What we weren't expecting to see was how active the prophage genome is in the lysogen. So what I have done here is I've circled the genes that were found on the phage genome that can be detected, their expression can be detected in lysogen cultures. So there's quite a lot of the prophage that's active in the lysogen culture. We've published this work, but one of the things that we demonstrated was that there were several gene expression patterns that changed drastically with the effect of prophage. And these were host genes, not prophage genes. So these are genes. Here we have Fimbria genes and the GAD response gene, which allows acid resistance in E. coli, that are really upregulated in the lysogen. And we also have malate quinolone oxidoreductase, pyruvate dehydrogenase, part of the TCA cycle, and cytochrome B oxidase, which is part of the aerobic respiratory chain, that are decreased. I don't want to go through this in great detail, but this demonstrates the GAD system. And the way the GAD system works is there are surface receptors that detect changes in pH that are responsible for upregulating genes in the GAD system. These genes in the GAD system also include glutamate decarboxylase and GAD-C as an antiporter. So what happens is this allows hydronium ions to be added here to glutamate to create GABA, which is then exported from the cell. And this allows the cell to deal with increased hydrogen ion inside the cell and enables the cell to deal with an acid response. Now, one of the things that was very important in E. coli 057 was that it was very acid resistant, much more so than normal E. coli cells, and it enabled an infectious dose of ingesting anywhere from 10 to 100 cells to cause disease. We went on to demonstrate that the expression of those genes and the control of the GAD system in the E. coli Shiga toxin phage lysogen was all down to the expression of a transcription factor, C2, in the lysogen. Now, this C2 is different from the C2 gene of lambda, and when it was expressed alongside C3, this enabled a drastic increase in the phenotypic ability to survive acid stress. So we went on to prove that it was the transcriptional activator C2, which is normally part of the control mechanism of lambda, was directly responsible for turning on the genes in the lambda lysogen that control the GAD system. And so here we have prophage control of bacterial systems in a way that provided a fitness benefit to the bacterial host. So we also looked at type 1 Fimbria. We knew that the Fimbria genes were upregulated and that these Fimbria genes had been tested and shown to have an effect on E. coli swarming phenotype. Now, we know that from our experiments that the flagella genes are downregulated upon prophage induction, but we demonstrated that the lysogens possess a swimming phenotype that the naive cell does not. So to do these assays and to interpret this, we have a spot of the lysogen, a spot of the naive cell, so this is carrying 24B, and this is the 24B prophage that has one particular gene knocked out just downstream of the C1 gene. And you can see here that the lysogen has a swimming phenotype that is not possessed by the naive cell, and that in the simple removal of the single gene downstream of C1 was responsible for removing this modal swimming type. So we found another gene that is in the process that we're in the process of doing further investigations on. I invite you to have a look at UAE's poster, which is being presented at this conference, for more information on what she has done with this information. So the take-home message again is a single gene in the phage genome is having an impact on expression of multiple genes that control distinct phenotype within the bacterial host. It would be interesting to see if very similar outcomes could be found with other phage host systems. And so I teamed up with Chloe James, Professor Chloe James at the University of Salford, and we, along with her colleague Ian Woodhead, and we set out to look in more detail in Pseudomonas aeruginosa, specifically in the Liverpool epidemic strain. This strain, 5 prophages, beginning with prophage 2, 3, 4, 5, and 6. Prophage 1 has been shown to be misannotated as a prophage and is a genomic element. We set up with the same idea. We set up growth conditions where we could limit the amount of phage that were being produced, and we set up libraries to examine gene expression through RNA-seq. And you will have heard a bit from Enrique earlier today, and you will hear from Ravathi Krishnamurthy tomorrow about work that they have done on understanding the impact these prophages have on the Liverpool epidemic strain of Pseudomonas aeruginosa. But what I'd like to point out to you here in these volcano plots is that comparing lysogen to induction conditions, this is the naive cell grown in LV. This is the naive cell grown in a different media. And what you can see is that there are significant differences in the number of host genes that are expressed in the lysogen conditions. Our Phage genes are colored here, and they tend to show up during the lytic replication cycle. So we have Phi2 being represented by blue dots, Phi3 by green dots, and Phage 4 by these orange dots. So what I want you to take from this is you can see that there are significant differences in the way the gene expression is occurring for bacterial genes in the lysogen cultures. There have also been some ways that we've started to look at this so this is a K-cluster centroid heat map, and the data are normalized to PaO1. And we can see that Phage2 and LB or M9, different combinations of these phages, cause changes to the expression patterns of les genes. And you can see that we have here what we expect from the single lysogens, but in the double lysogens, these outcomes are different. And so we have examples here of a phage having an impact on the expression of other phage genes or the impacts of other phage genes. So we've got evidence of crosstalk between the prophages and the way that they impact their bacterial host cell genes. Some of the systems that we have in prophage have an impact on deal with motility, some drug efflux pumps, alginate production, chemotaxis signal detection, metabolic pathways, and biofilm-associated gene expression. So these are all major players in Pseudomonas aeruginosa virulence. And here we are showing that the presence of the prophage has an impact on the expression of these key virulence phenotypes. So, in summary, lots of work to do yet. It's clear that prophages do impact gene expression of their host, and there is crosstalk between phages. And we were really excited to find this only a month or two ago. We're currently confirming phenotypes and testing phage genes for their ability to control these phenotypes. It's clear that prophages can impact their host beyond the provision of the toxage or carriage of other sort of single genes, often referred to as morons. Phenotypic alterations in the lysogens can and do occur. Phage genes have surprising secondary functions, as we showed with E. coli with C2 and C1. If you want more information on the externals of C1 functioning, please see Yui Chai and her poster. And the big take-home message here is prophages are not silent passengers in all of this. So we have now two prophage host systems, quite different from one another. In both cases, these prophages are having a significant impact on the phenotype of their host. And so I suspect that this is a very common occurrence, and that we are really only just starting to unpick some of the reason behind why we see prophages in so many bacterial genomes and why we see them in. I'd like to take this time to thank the organizers for allowing me to speak to you today through this recorded media. And I'd also like to acknowledge all of the people that have helped over the years on the data sets that have underpinned this talk. So work on the STX phage project, the RNA transcriptomics were performed by SCIA. And Yui Chai, a final year PhD student, is here presenting a poster of her work with C1 and gene 13, 24B. I also need to acknowledge the people and group that have worked on the Les Prophage project. These include Dr. Ravathi Krishnamurthy and Dr. Enrique Gonzalez Tutuero. And I need to let everyone in the audience know that these two people are absolutely wonderful researchers, and they are both in need of a job as our particular project comes to an end. So I would invite anyone to go and speak to them. Dr. Andrew Martin has been invaluable in this project from the University of Salford. Mrs. Reem O'Meara is joined us, and she is doing work currently trying to understand better Prophage 5, and she's presenting a poster here. And we've also been supported quite a bit by Mr. Haji again, who is a visiting student from China. And I also need to point out the invaluable help that I have received from my collaborators, Professor Chloe James and Professor Ian Goodman at the University of Salford. We obtained the funding for this project from the BDSRC to work on the Les Phages, and we are also collaborating with Professor Joe Fothergill at the University of Liverpool, who was involved in first highlighting the importance of Les Prophages in virulence of the Liverpool epidemic.